think it is strategically important for us to support the work that they are doing. I think everybody would agree that especially in the last year or two, the efforts from the SAMRC to battle the COVID-19 challenge has come to the fore significantly. But of course, it's much wider than that. I mean, you've always been involved in many different projects and initiatives that's important for health issues and mitigation in South Africa. So Brigitte, this is indeed a very welcome contribution for us as the CHPC to help you showcase, and of course for the CHPC to be part of the successes that you will talk about. Maybe just to indicate, she's the research program of Brigitte has been running, I cannot remember how long it is now, Brigitte, but it's probably at least the last 3 years or more, if it's not 4 years already that you've been actively leading this program. Sorry?
Yeah, we started in November 2018. We approached you for the first time.
Yes.
So it's a while. It's a while.
Yeah, it's a while. And I've just had a quick look, Brigitte, in the full time that the program has been running, about more than 1.4 million hours have been used already, and 235,000 just in the last 3 months. Now, the actual number is not that, may not say that much, but when it comes to bioinformatics type, say, applications and workflows, this is quite a significant number if we compare it to some of the other programs. So it just shows how active the program has been. I've just checked, there's 12 active members in the program. With active, we mean actually using hours and have produced work from the education. So that is also significant. And of course, there is significant outputs. I mean, I just know about that one very prominent paper that came out in Nature 2 or 3 months ago. So this is a high profile, and we are celebrating with you on the good outputs that we are seeing. So with that, I'm going to hand over to Brigitte. You see the bio and the abstract was shared, so I'm not going to say much more about this. Of course, the title of the talk is up there, Host Genetic Factors Contributing to Susceptibility of COVID-19. So Brigitte, with that, I'm going to hand over to you, and then to everybody, will be opportunity in the chat to ask questions as she goes along, and of course afterwards. So thank you very much, Brigitte. Over to you.
Thanks, Werner, and thank you for the opportunity. So the way I like to give talks is, if you have questions in the talk, stop me, because very often we forget as the talk goes on. So if you have any pressing questions that might come up immediately, stop me and ask me then. I have no objection. And then for the questions that I can't answer, I have Madlu Moller and Desiree Peterson with me, who can probably answer the other questions that might be logistics to the project itself. So, Werner, thank you for the introduction. Today, this will be an overview of the project that we are running in collaboration with the CHPC. And I'm not going to go too much into the results, because lots of those are going to be presented at the CHPC conference. And I don't want to take away from the value of those talks as well. So before I actually get started, I think we often forget the acknowledgements, and by the end of the talks, everybody's always tired and forgets who's involved. But I think it's important to note that this project would not be possible at all without a number of collaborators. And that includes clinicians, pathologists, it includes different institutions who have given both money, time, and I think most importantly, samples. And I cannot thank both the CHPC enough for providing the infrastructure, although I will hold Eskom wholly responsible for my frustration at the CHPC at the moment. But all of those, all those jokes aside, I think what's important to remember is that this host genetics project is not just one-sided. It is, I think, a true reflection of a collaboration that has come together, and has come together quickly, and has come together well. So, of course, now I have a non-responsive computer. Werner, are you trying to say something? Oh, there we go. Say something. I see your highlighted, but never mind. So we know that the SARS-CoV-2 is a beta coronavirus that hit the world, I think, a lot harder than we thought it would. And the problem with it is that for a lot of the time we didn't understand it or its workings properly. But what we do know is that the average person, once exposed to the virus, will start presenting with symptoms within 4 to 5 days. Now, we know, as in all cases with any kind of individual, there is inter-individual variability. So you'll have some people presenting as soon as 2 days after exposure with clinical symptoms, and some people even as late as 3 weeks after exposure. So there is this thing of, okay, we would expect people to start showing symptoms within 4 to 5 days, but no two individuals are the same. So this doesn't really help us much in a sense. We also know that given this inter-individual variability based on both the onset of symptoms all the way through to when they present with disease, we know we have asymptomatic carriers. We have individuals who present with mild and moderate disease and then sort of get over it pretty easily. We know we have those individuals who have severe disease, and then we know there is a subcategory of patients who are critically ill with the disease. And very often, we sort of try to lump them into these groups and say, well, it's usually your unhealthy individuals and they will get sick, and there they'll go, your risk of getting critically ill is much higher. So embarking on the study we did, we've actually found there is this inter-individual variability is very important, especially when looking at host genetics. So we also know that the risk factors are usually your age. If you have something like an immunodeficiency, so whether a primary or a secondary immunodeficiency, if you have liver and kidney disease, diabetes, we know is a mitigating factor, heart disease, the list goes on. But what we don't expect to see is a subgroup of relatively young and healthy individuals getting critical or severe disease. That's the sort of question is why, in the absence of sort of cofactors and comorbidities, why do we have individuals that are healthy and have probably had no serious disease? Why do they go on to developing the severe clinical phenotype? And what is the driver behind it? So lots of our interest has come from the small subgroup of looking at younger and healthy individuals. Why do they go on to develop this disease? So essentially, you want to say we're looking at a relatively extreme clinical phenotype in these patients. So the aim of this project essentially is to identify these host genetics factors. So the first question is there a host component to developing severe disease? And if so, why is there this extreme immune response in these patients? And can we actually then explain the pathophysiology of severe COVID-19? So what this means is that you can then put the brakes on the disease if you know a patient is likely to develop severe COVID-19. I think it's like many diseases. If we know the cause and we know what's the driver, we can sort of put the brakes on in the long term. So how are we going about this is we're collecting samples from a number of different sort of sources. Some of them are private and most of them are public as well. But again, I think what's important here is the fact that the private and the public have come and have decided, well, this is how we should approach it. We're taking standard DNA extraction and we're whole genome sequencing these samples at the South African Medical Research Council. And we're looking at whether or not there are variants that can be prioritized across these patients. So to date, aside from another patient sample set of MIS-C or multi-system inflammatory syndrome in children, which is something that will be dealt with in the CHPC webinars, CHPC conference, we have collected 36 young patients who have severe COVID, and we've actually sequenced 21 of them. And this is important because it's a good subset of patients, again, because you're looking at extreme phenotype. You wouldn't expect there to be a huge number of patients. If you were seeing a large number of patients, we'd have a much bigger problem than we already have. So for those of you who are unfamiliar with the techniques of next generation sequencing, essentially what we try and do is and in this approach for host genetic factors is we try and look at the entire genome of an individual. And so we take the DNA, we extract it, and we break it up into a lot of pieces. We sequence these short pieces, and then we try and put the puzzle back together, essentially. So by assembling the entire genome again. And the reason we do this is to see whether or not the genome of the ill patient, how different is it from the genome of the healthy reference, in inverted commas. There is no such thing as a good reference, but we then map it back to a reference genome and see, well, what are the differences between the healthy and the affected individual? So essentially, it doesn't matter what we do. What's important for us to look at is the fact that we get an output file. We need the CHPC computational infrastructure to manage these files. It's not only the CHPC, but you need an HPC infrastructure to manage these files. We make use of the CHPC because it is just simply the most convenient until we can generate our own analytical platform. But what we try and then do is computation is determine these variants, these differences between ill individuals and healthy individuals. So what's important for us in this whole approach to identifying variants that might be of interest in these patients is that we need to make sure that there is some kind of speed or some kind of sort of, if you want to call it, you want to essentially get the result as soon as possible. And the reason for that is there's a patient at the end of the line. We can't take 20 years to decide whether or not, number one, there is a genetic component, and number two, these are variants that should be targeted. It's difficult because no two patients are the same, but what you need to do is analyze the data as quickly as possible. Anything you get, you find, you put into the lab, and then report back, hopefully, to try and manage the patient effectively or patients effectively. You need that analytical pipeline to be stable. This is, of course, in the HPC environment, but you need to make sure that what you do is stable and reproducible, so that if I take the pipeline and I give it to another group doing a similar study, they will come up with similar results. It needs to be robust and it needs to be maintained. Now, obviously, you need it to be sort of interchangeable. So if you do start collaborating, you can give the pipeline to the next guy and say, well, this is what we did, see if it works on your data set, which remains important in the work that we do, particularly that is patient-centric. So some of the problems we have, especially with managing the data, is we are looking at 3 billion nucleotides now, and things we've come up with in this COVID genetics project is the fact that finding the right tools to identify the variants is proving a challenge. SARS-CoV-2 is so new, there are lots of resources, some of which are proving not so useful down the line. We also know that processing this data is not as fast as we had hoped, even though we tried to analyze the variants, because even once we've got a short list of variants aligned to a genome, you've got 6 million of them that you still need to work through and shortlist and prioritize. And often one of the biggest problem we have is there are good tools out there, but we can't actually get them, number one, or they're just simply too expensive to use. And that's something that we want to try and stop or get rid of, is the fact that we don't want to use tools that are inaccessible in other environments as well. Goes back to that thing of everything we do needs to be reproducible. So one of the questions I was asked by a clinician not so long ago, if this is so difficult, if looking at the whole human genome is so difficult, why do we do it? So I think it's fair to say we want to move away from this trial and error medicine where you've got patients who are treated symptomatically. So you need to think about it like two patients may well go in to see a clinician and get diagnosed with the same medical condition. They will then subsequently be treated with the same drug. One patient may recover, another patient may not. Said second patient may go on to develop an even worse clinical phenotype. And that is something we want to, through doing this research, is hopefully put the brakes on. So you want to take away this whole thing of, once you've got a good variant that can be identified in a patient, or a subset of variant that can be identified in a subset of patients, you can then hopefully target them and treat them effectively much quicker. It then also subsequently will bring down that burden on the healthcare system, because you've got less patients who have this critical and severe disease. So instead of then treating symptoms, you're treating patients where you know there is an underlying condition. And from previous research that we've done in TB, we know that lots of these severe phenotypes are genetically driven. And given COVID, it is possible to assume then that there is this underlying genetic condition, which may actually then drive the severity of the disease as it progresses, or as their viral load increases, for example. So we know that there are a number of diagnostic tools. And in the past, we used to use quite rudimentary things where we did like, cytogenetics, and only significant genetic changes could be seen. Today, we're doing molecular level testing where you're looking at an entire genome across, in individuals, and we can find all sorts of things. Some of them are actionable, some of them are not. But the fact of the matter is, we need to then say, well, will a patient, for example, if we have the genetic cause, can they be treated, is question number one. If they can be treated, how do we treat them? And what is the most effective way to treat them? And the last thing is then obviously to monitor the disease. Is, can we, by understanding a host's genetics, predict how they are going to sort of progress throughout the course of their disease, should they come up with a particular clinical phenotype. And by doing this, you can essentially preempt and hopefully, again, put the brakes on the disease to shift from mild to moderate symptoms to going to that severe and critical clinical phenotype. And essentially what we hope to do by understanding this is to some extent do predictive testing. So you want to then be able to find those patients before they are essentially affected, or when they are still asymptomatic and they get picked up and they get identified as COVID positive, that they essentially then get sort of earmarked and followed closely so that we can see again, can we treat them effectively as necessary? Most of our cases at the moment are in children, which is complicated in its own right, but they are the best to study because they're a small subset with, in lots of cases, very, very few contributing comorbidities. So you look at them and you can understand how does the immune system respond. Once we understand how the immune responses, you can then sort of translate that into the next adult set of individuals who will then look at these this clinical phenotype. But what we found with the whole genome sequencing of these children is that making sense of the next generation sequencing has been like building a puzzle face down. It's been a lot more difficult than we thought it would be, mainly because once we've done the alignment and we found the genes and we found the variants, shortlisting them and understanding them has been a lot more difficult because there are no resources yet or enough resources to help us filter through those variants of which of them are important. So some of our preliminary results that we have found, we are making use of something that the one of Jean-Laurent Casanova's group and the COVID what's it called? The HGI, the COVID HGI is making use of, which is the Human Genetics Initiative. And basically what they're doing is we are using something called the Gene Damage Index and looking for genes and variants that are associated specifically with immune system pathways. So what we're trying to do is find genes and look at the clinical readouts and see, do those two correspond? But we still have a significant number of variants that get shortlisted, which are not always easy to look at in the lab. So we have now decided to look at essentially a three-phased approach, which is your adaptive immune system. So how does the adaptive immune system respond based on all of the receptor signaling pathways? The innate immune system, obviously the neutrophils, and as we've seen in many cases, neutrophils are often the first line of defense to many infections. So neutrophils will often be one of the ways that we move to. So that's been a focus of the study. And then, of course, cytokine signaling pathways, because many of these patients get massive cytokine storms, which you then have to try and control. And once you've controlled this cytokine storm, you can then hopefully bring the patient out of that huge and severe and critical infection. So what has been a challenge to us has been, so we've got variants and we've got genes, 5,000 of them on average per patient. Which of them do we shortlist and which of them do we prioritize? To date, there are very limited tools that can help us with that. And there are a subset of genes that we can look at, but in the African population, are these relevant, which is another confounding factor. So in summary, we hope that this data, well, actually we know that this data will contribute to COVID-19 research and the Host Genetics Initiative. But what's important is that this comes from African populations. It's not just European and not just where a lot of money is being invested. It's locally done, number one, and number two, it's focused on our population where it's relevant. We know that studying different populations with genetic causes possibly behind them can increase our understanding of disease, and understanding the pathobiology of the disease is incredibly important. And we know that once we understand the pathogenesis and disease outcomes, not only can you try and put your thumb on the way this disease is going to go, but you can identify individuals who are at risk, especially genetically, to developing severe disease. And then in turn, stopping this healthcare burden where you've got this overrunning of the ICUs and the hospitals and putting the brakes on everything. And I think a pretty grim outcome for a lot of these patients. So this is where I'm going to leave this talk. I am happy to take any questions. I've got the two seniors here who will take the questions that I can't answer. But I know that Crystal will disclose some of the results we found in her CHPC talk as well. I didn't want to take away from that, which I think is important. But yeah, I'm happy to take any questions you may or may not have.
Thank you very much, Brigitte. This is indeed a very good overview, and I can see there's many things that's being done, and I cannot claim to understand all the intricacies of what you are talking about, but I'm sure we have a very informed audience, and I can maybe just say to the audience, you are welcome to pose a question, either as typing it or put up your hand, and we will unmute you. Benjamin on the call will unmute you, and you will be able to pose the question directly. But as you talked, Brigitte, the one question, and I think this is something that many of us have asked a lot of times already, and of course, as I understand what you're doing, it is focused exactly on this question of why do certain people get affected by COVID so severely, whereas the next person that seemingly has no difference in terms of health or so does not get affected at all. And I suppose this is one of the key questions, because I suppose if you can identify that part of the population that will be affected the most, you can narrow and focus on, say, the vaccination of those individuals before others. I mean, do you have some answers for the ones of the people like myself that do not understand the genetic factors and so on? So well, do we know why? I mean, this is such a perplexing thing to me.
I think, Werner, if we could answer that question now, we'd all retire. I think, and a lot of us would be without a job. But no, we don't. So we know also clinically across the spectrum, the patients present very differently as well who have critical or severe COVID as well. So it's like not two patients will say they have the same cytokine storm. And if any of our clinicians are on this call, please step in. I can't see any of them. But if we do have, no two patients seem to react the same way. But why? Our question is driven by why do you have a healthy individual with no comorbidities who develop this massive bout of COVID, who either turn around literally on death's door, or it's a case that they don't make it. So that's the question. So I would, honestly, if we could answer that, we'd all be out of a job, I think.
But I can see the focus is very much on this. I see there's a hand from Ricardo Harry. Ricardo, you're welcome to pose your question. You should be able to speak. Otherwise, Benjamin, you can just check that Ricardo can talk.
Hi, good day, everyone. Hi, good day, Brigitte. I'm Ricardo. I'm also part of the CHPC team. I've read many years ago that, for example, men, the older they get, the better the genes they actually gonna be able to transfer to their offspring. The older they get. So it's like I've learned that genes are actually learning certain things as life progresses, as we get older. And the more they get used to our ways or our nature of progressing through life, the stronger those genes become. It's like the older or the genes that does not really matter sort of die out, and the stronger genes will prevail as we get older. I can't remember where I read the article. So I was just thinking, if we look at elderly people, especially the ones that prefer to have that healthy lifestyle, continue to keep fit, eat normal, healthy, have a healthy diet, does that contribute to maybe, or could it possibly contribute to maybe the futuristic fight for most diseases, especially now like COVID? Let's say, as young people, if we continue with that type of attitude towards our life and living it healthy, continue to enhance it healthy, good genes, does it maybe have any impact on our fight against disease, especially in this case? I think that's my question.
So Ricardo, I think what's important is that we are born with a set of genes, and we've all got the genes. There's often this misconception that, You know, some people have a set of genes and another people have another set of genes, but we've all got the same sort of breakdown. We've all got the same building blocks, I think is very important. So what we do over time is get variations and adaptations, and there's things called epigenetic modifications that we go through as well. So things do modify, but essentially the backbone doesn't change. And, you know, Marlo, I don't know if you've got anything further to say. So I think basically there are certain, we do know in some diseases that there are susceptibility factors, for example, which may make you more susceptible to developing a disease, and some protective factors which you may, you know, sort of inherit over time that then prevent you from developing certain diseases. But I'm not sure about that. I'd be interested if, I mean, I know you said it's a few years back, if you could refer me to the article that I could have a look at it, maybe just to get a better understanding of it. Yeah, that would be, I don't know if Marlo or Des want to step in, but yeah.
Yeah, I think you had that question at the beginning. Sorry, I'm ugly, guys.
Sorry, she is
asking about if, you know, the genes in men get stronger and that they transfer to their offspring. And I think maybe we're talking there about natural selection. So that's where we tend to inherit the genes from our parents because they had a survival advantage. So maybe in a few years, we'll be able to look back and see that those individuals who had COVID and passed away at a young age and didn't have children, that we're selecting against those genes in the human population. But we won't be able to see that now. And hopefully we'll contract that in a few years. So in human genomes, we do see signs of selection.
Yeah.
So for other diseases that people had, that had a huge effect on the number of humans around. And yeah, I hope that explains the question a bit better. Or the answer.
Thank you. Ricardo, do you want just a follow-up comment? I see your hand is still up. Otherwise, we can.
Okay, sorry, Thijs. Then one of the things which was also evident, also coming back to the gene or adaptation, was I do a lot of charitable work outside of academia and outside of my work itself. And we work with a number of groups of vagrants, for example. And we noted that a lot in the past 2 years, a lot of these vagrants was never immunized against COVID, and not a single one of them succumbed to the disease over the past 2 years. And they were some of the people that are most exposed when it comes to just daily living. And that was very interesting. Is it because of a stronger immune system? Because they are so exposed to so many things that we still have a lot of those groups when we go and visit them that are still there. None of them succumb to this disease. So maybe it's just another follow-up question based on that. Is it maybe the way they also genes or part of the genes adapted to this whole environment? That's my just an observation. Okay.
So I think in humans, we've got this wonderful immune system. It can adapt to things. And although the coronavirus disease 2019 is new, we know there are other coronaviruses out there. So maybe some people have been exposed to certain strains, and that has given them some measure of protection against the current one. We don't know that yet, so scientists are still working to determine that. And then, you know, some of those, so you were mentioning vagrants specifically, as you called them, and maybe they just did not get tested, or they just had mild symptoms, but as you said, not severe disease. So that would be a very interesting group to follow up on. I have to punt our research study here. We are collecting from the general public. So if any of you guys want to take part in the study, you can contact me or Desiree Peterson, and I'll ask Vanna to send out our details later and some slides. And then we'll send the buccal swab to you. It's a simple swab in the cheek that you do 30 seconds on each side. And we collect your DNA this way. And we'll ask a few questions about the symptoms you had, if you've been vaccinated, and if you want to share your genetics. So we are trying to establish a large cohort of participants. We want to go up to 10,000 South Africans. We've now started with the first 1,000. So Brigitte was just mentioning the severe COVIDs that we've sequenced, but we've got more samples back that are waiting for sequencing. So we are recruiting severe hospitalized patients from a few hospitals, but then also from the general public, because these are the categories we want to compare.
Okay, that's interesting mentioning that. So yes, certainly we can send that message to all participants here to get more information. So thanks for that. Thijs, your hand is up. I will get you the questions in the chat just now, but Thijs, you can pose your question so long. Thanks.
Okay, thanks a lot. I don't know if I've missed it, but are you testing people that didn't have COVID and the ones that didn't have severe symptoms to compare to the ones that had were severely affected by COVID?
Exactly.
Yes.
So that's our comparative group. So we have a few categories that we're collecting, and when you answer the questionnaire, the questions guide you through that. So we're definitely interested in people who never had symptoms, who never had to go for a test. Perhaps you had it, but you were just never symptomatic. For our active cases, we just include people who tested positive. So you had symptoms, you went, you got a positive PCR test. Yeah, so we have a few questions for that. And then,
okay, so you haven't tested anyone that never had symptoms or so far?
we've got,
Or maybe identify if they might end up
being
resistant. Being resistant. That's what I mean.
yes,
Because
so,
I've seen in families
yes,
where 6 out of the 8 of the family gets COVID, test positive. Doesn't even some of them get severely ill, some not, and then there's 1 or 2 members in the same family that had the same exposure, but they tested negative, nothing happened to them. Maybe you should look at those as well to see
exactly.
maybe what makes them different.
Exactly. We are asking those people to come forward. For example, I know of some, and I think we all know someone who actually gave miles to a COVID patient and got no symptoms, never tested antigen positive even after all that. So they are definitely interested. In genetics, we refer to them as these genetic resistors. We also see them when we study TB patients. So people who live in a high TB incidence community, but never get sick and never get infected. So they're definitely a group of interest for us.
It just shows how many of these intriguing questions we still cannot answer. So that is interesting. I'm just going to go to some of the comments and questions in the chat. Brigitte and Marlo, Alexandra there just says very interesting and thanks. We'll look forward to the findings at the CHPC conference. I will say something at the conference just now. And then from Yolandi Swart, thank you for your information. Help me understand where we're going with this and pharmacogenetics, precision medicine. And then Tracy Euster, she's asking, should we shift our efforts towards the investigation of tools utilized for variant prioritizing, considering that everything up until that point in the pipeline works rather efficiently? So maybe you can just respond to that, one of you.
So Yolandi said, where are we going with this in terms of pharmacogenetics, or genomics? So Yolandi, the idea would be then, of course, if you have the gene, you can then, predict, then you'd obviously have protein expression, for example, and you'd be able to look at it functioning. And then from there, you could ideally then either target the protein with a particular drug, for example, or, if it's an immune system dysfunction, you could then do it that way. But the idea would be then, if you have a patient who is severely affected and we know what the gene / the variant is, you could then manage and treat the patient accordingly. So instead of throwing, let's say, for example, I'm just using a broad spectrum antibiotic as an example, it's like then having rather than having the bug typed, so are you looking at gram-negative, gram-positive bacteria, for example, but this time you would then say, well, would the patient be able to be managed differently and treated differently? So we know, like, for example, in our TB patients, if they have an interferon gamma receptor 1 mutation, for argument's sake, should they be on INH prophylaxis, for example, then you can treat them that way, if it makes sense. Or should you vaccinate a patient, or is it maybe advisable not to if there's something else? If we've seen a particular mutation, then drives on to very severe disease. So that is the question. And then if you are happy with that, Yolandi. Tracy, should we shift our efforts? Tracy, the short answer is yes, but because the field is not static, it's very difficult to choose a toolkit and a pipeline that is actually effective. So what we found is that there are lots and lots of good tools out there, but each one of them sort of seems to perform a different function. So we actually have in-house methods that we use to prioritize variants, which seems to work quite well for us to date. But I mean, it changes all the time. So yeah, if I could find a way to make use of all the databases that are out there to prioritize the variants, I would. But at the moment, there's just too many. And the ones that are out there, I mean, we've actually found that are quite expensive to make use of. They're effective, but they're very expensive. So what you end up doing is, for example, merging all your VCFs and then only submitting one file because you can't afford the $250 to run one sample when you want to make this a big study of 10,000 participants. So,
and I think to add to that, you know, in the end, we always have to do functional verifications. Yeah. This is just a predictive tool, and they're great, but they're not infallible.
Yeah, and we've often, in our own patient subsets, again, going back to our TB work, we found variations in genes that are predicted to be pathogenic. But when you go and do the functional work in the lab, actually, it doesn't seem to have as big an effect as we thought it did. Yeah.
And it goes the other way as well.
Yes, it does go.
And maybe we're just not stimulating the right things in the cell culture, but
yeah,
there's all those levels of complexity to add.
Thanks for those responses. There's a next question. I find it very interesting from Caitlin Cutler. She's asking, how do you think the interindividual variation could affect the efficacy of the vaccines? And there's also a question that I have. I mean, is it the case that there will be certain individuals for which the vaccine will simply not work? I mean, because of the different ways we are put together. But that's an interesting question. Maybe your thoughts on that?
I think we've already seen, we've all heard stories about people who had the vaccine and then still got severe disease. So you will always have the outliers that we've been discussing, right? And in some people, we've now seen that the vaccine wanes over time. That's why we need boosters. So perhaps this will happen sooner in some people, that they'll have to get a booster sooner than others. And those are all very, very interesting questions that we just don't have the answer to yet. I feel like I'm on repeat here, but I think it's because it's new disease that we're still learning this. And this is, I think, where personalized medicine comes in. You know, we keep talking about it's going to happen, and it's happening in some realms, but there must be a way to roll it out to a bigger audience. I was on a call the other day about the pharmacogenetics of TB, and one of the clinicians said, well, we'll never have personalized medicine for TB because, you know, the TB program that we have at the moment is working well by rolling it out, as Brigitte pointed out, on the global scale. And I think this is what we should still be aiming for. In the end, we have to prioritize the individual and come up with a reasonable treatment scheme for someone else, for everyone.
Yeah, no, I think, don't apologize for not having all the answers. I think we are all appreciating it and under no illusion about how complex the challenges are here. So I think we all appreciate the efforts and the results that we do or are obtaining. So we are all understanding of this. Ricardo, I see your hand is up again. You are welcome to pose another question.
Yes, I've got two observations again that I also made during. I'm an avid gym goer. I go almost regularly. One of the things that I've noticed was when people come to the gym and they haven't exercised maybe for a week or maybe 2 weeks, and they hit it hard. I noticed that there was a few people that I personally know that actually contracted COVID, although they were really fit. When they come to the gym, they hit it very hard for maybe the 1 or 2 days, and then they become susceptible for contracting it. That's the one observation. The other one is that is now being away from exercise for about a week or two. The other one is I knew about 3 healthy individuals almost look like bodybuilders that also succumb to the disease itself. So my question is, does performance enhancing drugs impact on your ability to contract the disease? Not saying that these people use those drugs, but is there possibility like the excessive use of steroids or any other performance enhancing drug, does it impact your ability to fight the disease? Because I know in the medical field, we also use steroids for certain, to fight certain diseases. And sometimes it might have a counter or productive effect on people that want to try and get rid of the infection itself.
Yes, you're right. You know, anything that you're taking that's not under the supervision of a medical doctor can likely affect you. And as you know, steroids, they will have an effect on the immune system. So it's very possible that it could have an effect, yes.
Thank you very much, Brigitte and Marlo. So I don't see any more questions on the chat. If there's anybody that wants to put a question, now is the time so that we can still deal with, or put up your hand and we can ask Brigitte and Marlo to address it. I just want to highlight, and it was mentioned a few times, the CHPC National Conference. I've put the link there. If you're not registered yet, please proceed. This is a fully virtual event. It runs from the 1st to the 3rd of December. So we're basically less than a week and a half or a week and a half away from the start. The program is basically finalized. You can see all the different speakers and talks there. And I think it is appropriate to highlight that Marlo, that is joining Brigitte here, Marlo Moller, Professor Marlo Moller from Stellenbosch University, will be one of the speakers in one of the sessions to take this topic further. So Marlo, we are very appreciative of you accepting to do the talk and bringing this, let's say, discussion focus to the conference as well. So we hope that we have an even bigger audience there to hear what you are doing and maybe also to pose the questions. So yeah, so the invite is open to everybody. Please go and register and attend, even if you can only do a few sessions. It is free and it will be easy to attend. Okay, so I do not see any more questions. So maybe I can just end off and thank Brigitte and Marlo for your participation today and telling all of us a bit more about this important work. As I said in the beginning, I'm just very pleased that the CHPC is playing a part in this and that we can help to advance this work and this research. So we will continue to do this, and whenever we need to do something more or something else, you know, we are ready to do this. And yeah, thanks for also to show this to everybody in the audience. Thijs, I see you have a comment to make still. You want to come on? Am I understanding this correctly? You're welcome. Thijs, you should be able to speak if you want to say something.
Oh, okay, thanks. I want to make a comment on Ricardo's comment where he said that someone that did the gym hard after a few weeks off, they tend to be sort of more susceptible to that. I have found in my own, when I was still gymming a lot before I moved out to the farm, that if I've been off for a week or two, and I go to the gym and I start too hard the first few sessions, I tend to be more susceptible to colds and flus as well. I think you're overstressing your immune system in that time, and that makes you more susceptible. And I think it's probably something like that as well with the COVID, people getting it when they eat the gym too hard after a time off. That's all.
Thanks,
I wanted to say.
Yeah, that's an interesting observation. Ricardo, I see your hand.
Okay, then the other observation that I also made was, if you have two healthy people, perfectly healthy people, and they exactly have the same physiology, genetic makeup, everything, what I've also noticed is a person that has some sort of more strenuous environment that they're exposed to on a daily basis are also more susceptible than the person that doesn't stress about, let's say it's twins, that doesn't stress about these things, And in fact, in this case, I actually saw in an environment where one twin was more susceptible to picking up these colds and flus and COVID related because of the nature of their work and the stress that they were exposed to. Whereas the other one, although they lived together, was quite cool and didn't really show any symptoms sometimes. And it just got a bit of a tranquil environment that they work in. One is a janitor, one is a train driver. So I noticed that the one was very susceptible for picking up disease. Is it because maybe of the stress that the body actually on a daily basis have to cope with, or and does it impact your? It probably will impact your susceptibility to pick up viral infections or other disease, for example. Is that also part of? I actually got a bit late into the presentation, but is that also part of what you measure?
So we do ask questions on the environment of study participants. You were using the example of identical twins. So in our cohort, I can tell you we had identical twins, where the one, and I think you might have seen this in the U magazine also, they're not the same people, but I've seen more than one example. So they've got the identical genes. Yes, the environment might differ, but both twins passed away from COVID-19 weeks apart. So that also gives some support that there's a genetic influence contributing to it. I think, if you're talking about general flus and colds in twins, those diseases have been around with humans for a long time. So our immune systems have evolved to protect us against it. And yes, there are things like your personal immune status at the time might play a role if you're getting sicker or not, or having stress. I see in the comments also someone saying some psychological components. Yes, we are not microbes in a little container, all the same, being treated the same. We are individuals experiencing different things, and that's why we have different outcomes. So we are not just the sum of our total genes. This is also influenced by our environment and our experiences. So I don't know if any of you've seen that movie Gattaca. It's a very old movie about genetics and genetic discrimination. And even there it comes into play. Your genes is just a part of who you are. And yes, it plays an important role, but it's not the be all and end all.
Yeah, thanks, Tracy, also for your comment. You're saying the aspect of epigenetics comes into play. Okay, but thank you very much for the participation of the participants. We appreciate the interest, and I think Marlo and Brigitte, you can see that this was really a topic that people were engaged in today, and thank you for sharing your insights and answering the questions, and of course presenting the talk. I can just mention to everybody, this talk is recorded and it will be available on YouTube and so on, also for people afterwards to view. So thank you to the two of you for contributing to this as well. So I think with that, we will end it for today, and thank you, and see you all at the conference. Thank you very much. Keep well. Have a good day.
Thank you. Bye-bye.
Bye.
Okay.